So what I want to do is I want to give you an overview of a system that I've been working on, at least the Quorum-Sensing part of this, for probably 15 years or so. And as I have worked on this, I have spent particular interest in developing or trying to understand the biology of this system. So what's going to be running through this whole talk is the biology of what this is all about with respect to the organisms that I find particularly interesting. So, Vittoria told you the title, and I want to introduce you to a family of bacterial plasmids, and these are plasmids that are turned out to be very important in the Alpha-Proteobacteria. They're a family of plasmids. They are based on a particular replication system called the Rep-ABC replicator system. It's a series of 3 genes, Rep-A, which is a regulatory gene, Rep-C, which is the DNA initiation protein. The ORI for replication lies within the Rep-C. We have not studied this in any great detail. People like Steve Winan's lab and also a lab in Cuernavaca, Miguel Ceballos have studied this extensively. But it's an extremely interesting replication system because the DNA elements with which this replication system are associated first are very stable. These plasmids are low copy number. They replicate in synchrony with the chromosome, and they are broad host range with respect to the Alpha-Proteobacteria. So you can take a plasmid from one Alpha-Proteobacteria and put it into another Alpha-Proteobacteria, and they seem to work quite well. The really interesting and important biological characteristic of these replication systems is that they have all of these traits, yet that they can carry very large amounts of DNA. All right? And this just gives you an indication of what was known back in 11 years ago on the phylogenetic diversity of these systems. And what you can see from this is that the Rep-C gene, which is the important gene for replication and also for incompatibility, is evolving like crazy. So there are many, many examples of this replication system. Some of them are incompatible with each other, and many of them are compatible with each other. So you can put many of these plasmids together in the same cell. All right. The importance of these replication systems is indicated by the fact that sometimes these plasmids or the DNA that is carried by these replication systems define what these Alpha-Proteobacteria are. So for example, in pathogenicity, we have the TI plasmids of Agrobacterium, which you will hear a lot about, the RI plasmids of Rhizogenes, chromosome 2 of Brucella. These are all of plasmid origin. Symbiosis and nitrogen fixation, I think you'll hear a little bit about this from Pete. Cynorhizobium meliloti, 3 species of Rhizobium. All the symbiosis functions are all carried on a Rep-ABC type plasmid. In addition, some of these plasmids in other less well-characterized organisms encode interesting features. For example, Oligotropha carboxylivorans, its ability to utilize carbon monoxide and carbon dioxide is carried on one of these Rep-ABC plasmids. All right. I will come back to this idea of opene utilization a little bit later. All right, just to give you a feel for what genomes look like in some of the Alpha-Proteobacteria and how these plasmids contribute to this, I list up here a number of members of the Rhizobiales, and I show you the primary chromosomes of these bacteria, and then all of these elements that are circled in red are plasmids or chromosomes that carry and replicate from Rep-ABC type replicators. So for example, Int'l has a primary chromosome, and then it has 6 plasmids. Each one of them replicates from a Rep-ABC type replicator. Agrobacterium, Radiobacter, and tumefaciens have 2 additional plasmids, but also they have a second chromosome, all right, here and here, and that second chromosome is a Rep-ABC plasmid. So genes are moving from primary chromosomes to these large Rep-ABC type plasmids. In Radiobacter, it remains a circle. However, in the BioVar1 Agrobacterium tumefaciens strains, the secondary chromosome, which replicates from one of these Rep-ABC type systems, is a linear molecule. All right. So the red indicates Rep-ABC type replicators. It's not restricted just to the members of the Rhizobiales, but in Brucella suis. In fact, in all of the brucellas that have been looked at, chromosome 2 is always a Rep-ABC type plasmid. In Spheroides, it's also a Rep-ABC. This is the first second chromosome ever discovered in bacteria, by the way, by Sam Kaplan. And in several other bacteria that have interesting traits these plasmids predominate. So this is an extremely important replication system. Okay, so many of these rep-ABC elements are conjugative. They can transfer from one bacterium to another. The generally conjugative transfer systems in the gram-negative bacteria are composed of two types of functions. There's the DNA metabolism functions, which Eric Lanka has called the DTR systems. These are composed of the nicking enzyme and the factors that work with the nicking enzyme to cause the single-stranded nick at the origin of transfer. And then the second component is the mating bridge. And in gram-negative bacteria, in most gram-negative bacteria anyway, this is by and large a type 4 type secretion system. So amongst the rep-ABC plasmids, many of them are conjugative, and if they are conjugative, they oftentimes fall into two classes. So here we have just kind of the prototypical rep-ABC unit here, and the two classes of replication system, of conjugation systems are phylogenetically different, sort of. All right. So if you look at the mating bridge system, the MPF system, both of them have a type 4 secretion system, but this type up here has what's called a TRIB type system, which is very similar to the mating bridge system of the INC-P1 plasmids like RP4. And down here, the other type of system that you find is the VIR-B-like system, which is very similar to the TI plasmid type VIR-B. All right. Although this is not the virulence type 4 secretion system. This is a conjugative type 4 secretion system. Now, amongst the DNA transfer systems of these two types of transfer systems, they are similar but different, all right? And so if you have a TRIB type 4 secretion system, then you have this type of DNA transfer system, and if you have a VIR-B type 4 secretion system, you have this type. The only place they really differ is in this gene right here, TRA-F. And the reason that they differ here is that TRA-F is a protease that's involved in processing the pillin protein of this particular type 4 secretion system. This is different, so it doesn't need that gene. So just to give you an idea then, here is the DTR, the DNA transfer systems of the two types of transfer systems. And so you have the nicking enzyme here, you have the relaxosome proteins here, the coupling proteins here. The ORI-T, the region for conjugative transfer, where it starts, is located here, and they're very closely related to each other, these two type systems, right? And not only that, but they look like INC-Q plasmid ORI-Ts. And down here are the genes that comprise the mating bridge, the type 4 secretion system. And there is, although there are relatedness between the genes between the TRIB type and the VIR-B-like, there are also differences in the organization and the composition of these. So we have two really phylogenetically different transfer systems that are evolving independent of each other. I put this up because I kind of like this idea of the rep-ABC system, and that is that the way that I look at it is that the core unit of these large plasmids and sometimes secondary chromosomes, they are like the tractor part of a Mack truck. All right, so this is the rep-ABC system. It can replicate itself. It can also transfer itself down the highway. All right, but what it's really good at doing is carrying lots of freight. And that freight can be chromosomal genes, it can be TI plasmid genes, it can be SIM genes, it can be anything that defines that particular organism. All right, so in the transfer systems that these plasmids have, oftentimes transfer is regulated. And the regulation then is at the level of transcription. All right. For the sake of our discussion, though, we're only going to discuss the regulation of the TRIB type 4 secretion systems, and that is the ones that are exemplified by the TI plasmids of Agrobacterium. So we'll spend a lot of time talking about this. So you all know this. So this is not a phylogenetic tree. This is a tomato plant. And this is a tomato plant that has been infected with Agrobacterium tumefaciens, and of course you get these crown gall tumors on the side of the plant at the infection site. The reason that the bacterium can induce these tumors in the plants is because it has a plasmid. And these plasmids are called TI plasmids for tumor inducing. They have things that plasmids have to have. They have a rep-ABC type replication system. And then they have a series of genes that are involved in inducing tumors. One set of genes are carried in what's called the T region. This part of the plasmid is processed and transferred over to the plant, where it finds its way into the nucleus of the plant, integrates and expresses genes, turning normal plant cells into crown gall tumor cells. The genes that are involved in processing the T region and transferring it to plants are encoded in the Vir region, and really this is nothing more than a modified conjugative transfer system. All right? All right, so characteristics of crown gall tumors. Well, first, they are chaotic, and they grow in an uncontrolled fashion. They grow without additional hormones, so you can culture plant tissues in tissue culture. And normal plant tissues require two major hormones for growth in tissue culture, auxin and cytokinin. Crown gall tumor tissue doesn't require either of them. They're graftable. This is the most important part probably. The crown gall tumors persist in the absence of the inducing bacterium, and what this means is that it's been a permanent genetic change to the plant cells following the infection by the bacteria. Because plants don't have the right kind of a vascular system, these tumors are not metastatic. They have one other characteristic that is going to be quite important for what I'm going to talk about, and that is they produce novel carbon compounds, and these novel carbon compounds go by the generic name of opines. So we're going to focus on opines for a bit. All right, so this sort of gives you the cartoon of what this is all about. I draw this to scale with respect to who I think is the most interesting and important part of the interaction. All right, so the plant is small, the bacterium is large. The bacterium receives signals from wounded plants, induces the Vir genes, processes the T strand. The T strand is transferred over to the plant cell, where it integrates into the nuclear DNA of the plant, and then that tells this plant cell to turn into a crown gall tumor cell. So that's the main idea behind that. Okay, I mentioned to you that crown gall tumors are characterized uniquely by the fact that they produce these novel low molecular weight carbon compounds called opines. So opines fall into about 8 chemical classes that we know about. The class of opine that a tumor will produce is determined by the TI plasmid that is present in the bacteria that induce the tumor, because the genes for the biosynthesis of opines are located on the tDNA. So they come from the bacterium. All right, so this gives you an idea of what some opines look like. This is the family of mannitol opines. These are imine conjugates, in this case of glutamine or glutamate, and mannitol. All right, and so we can have several of these, and these will all be produced by the crown gall tumors. Okay, so the roles in the opines in the biology of Agrobacterium tumefaciens, it turns out that the opines are there for 2 reasons. The first reason is that they are specific sources of nutrients for the bacteria. So Agrobacterium engineers plant cells to produce opines by transferring to the plant cell's tDNA. The value of the tumor genes, that is the genes that make the single plant cell grow into a tumor, is that now instead of having just one plant cell making opines, you have thousands of plant cells making opines. That is, you have a factory for making opines. So the bacteria then can take advantage of those opines by utilizing them as sole source of carbon and nitrogen. And it can do that because elsewhere on the TI plasmids are the genes for opine catabolism. So the TI plasmids are actually binary with respect to this. They have the tDNA that tells a plant cell to become a tumor and to make opines, and they have a set of genes that tell the bacteria you can take up these opines and utilize them as carbon sources. All right? Okay, and so that's depicted here. Let me go back. The second role of opines is that they not only act as nutrients, they act also as signals. And so they are the specific inducers of the TI plasmid conjugative transfer system. And this is what we're going to focus on. All right, so now if we expand this model then, we have the bacterium inducing tumors. The tumors are now making opines. The opines are feeding the bacteria by virtue of transporters and catabolic systems also coded for by the TI plasmid, so the bacterium can divide. All right? But in addition, the TI plasmid can transfer itself by conjugation from one bacterium to another bacterium, and that conjugative transfer system is induced by the opines that are produced by the tumors. Okay? What this results in then is another bacterium down here that now has a TI plasmid, and if it's a suitable bacterium, it can utilize the opines that are produced by the tumors and now is a competitor for this bacterium up here. All right, so just to give you an idea of what the transfer systems of the TI plasmids look like, Remember, we have two types of functions. We have functions that have to do with DNA metabolism, the nicking at RET. Right? Those are coded for by a set of genes called TRA, and they're located down here. I showed you those before. Here's the nicking enzyme, relaxosome proteins, and then the coupling protein here. And in addition, we have the mating bridge. We have the type 4 secretion system, and that's coded for by an independent operon that's located usually elsewhere on the TI plasmid up here. All right, so that's what the conjugative transfer system of this type looks like on these kinds of plasmids. We see this on TI plasmids, we see this on RI plasmids, we see this on opine catabolic plasmids, we see this on symbiosis plasmids of rhizobium, we see this on the CO utilization plasmid of oligotrophic carboxylivorans. So this is a widely dispersed way of organizing conjugative transfer genes on these Rep-ABC type plasmids. Okay, so in the TI plasmids, then, opines induce transfer. And this is a slide that's just designed to show you that. We're taking our favorite strain, and that is a bacterium that has the TI plasmid called PTIC58. So this is our donor. We are going to mate it with a recipient that lacks the TI plasmid. We can do these matings directly on selection media, all right? And so we take the wild type strain and we either grow it in the absence of this opine here, or we grow it in the presence of this opine, and we ask, will you mate with the bacteria that are present on the petri dish? And what you can see is that if you grow the donor in the absence of opines, you get essentially no transfer of the TI plasmid. If you grow the plasmid, the strain in the presence of the opines, then you get lots of transfer. And in fact, transfer frequencies approach 10 to the minus 2 per input donor. So it's pretty efficient. Opines induce it quite nicely. Now, I lied to you a little bit. Sometimes you get a colony or two when you grow the donor in the absence. And what you find is that these guys are transfer constitutive mutants. So if now you take one of these colonies and you remate it, what you find is now it transfers at a high frequency no matter whether you grow the donor in the presence or absence of the owner. So these are mutants. And this led us to the discovery of how this whole thing is regulated. Okay, so it turns out that the genes that are involved in conjugative transfer, remember the DNA processing genes here and the type 4 secretion genes up here, are generally strongly off in the absence of the conjugative opine and strongly on when the conjugative opine is present. They are strongly on because they are controlled by a transcriptional activator called TRA-R. This is a member of the Luxar family. This is why I'm here. All right? So TRA-R then activates expression from promoters that are located here and two divergent promoters that are located here. But remember, since TRA-R is a transcriptional activator of the Luxar family, it is going to need a ligand. It's going to need an acyl homoserine lactone ligand. And that acyl homoserine lactone ligand, like is in many but not all cases, is produced by these particular bacteria themselves. It's produced by these bacteria because there's another gene that codes for a synthase. That gene that codes for a synthase, it turns out to be the first gene of the type 4 secretion operon, that is TRA-I. So in the absence of the conjugative opines then, all of these operons are shut down. But nothing is completely shut down ever. All right? So the bacteria are making very small amounts of the acyl homoserine lactone agrobacterium autoinducer. All right. In the presence of the conjugative opines, the system turns on strongly. TRA-R is made. All right. The acyl homoserine lactone accumulates at basal level activity levels until it reaches a critical concentration, which we will talk about. It interacts with TRA-R, drives dimerization and stability of TRA-R. TRA-R can now activate transcription, and the conjugative transfer system is turned on, and the TI plasmid can transfer. Dependent upon opines, that means it depends upon the bacterium being associated with a crown gall tumor. Okay? All right, so that's the acyl homoserine lactone. I guess I don't have to tell you anything much about that. So now we have to ask the question, what's the role of the opines? So how does the opine fit into this whole story? Remember, you got to have the opines to turn on the system. Well, it turns out that for the TI plasmids, it's very simple. In all cases that have been studied, TRA-R, which is this gene right here, is a member of an operon that is controlled by a transcription factor that controls other genes in response to the opines. So how can we make that clearer? So on this particular TI plasmid, PTIC58, the conjugative opene I showed you was the sugar phosphodiester opene called agrosynapine. So agrosynapines are taken up and utilized by agrobacteria that have this type of TI plasmid because they have a group of genes right here that code for a transporter and 2 catabolism genes for the uptake and utilization of the agrosynapine openes produced by the crown gall tumor that this bacterium has induced. All right? These genes are regulated. They are regulated by this repressor, ACCR. This repressor not only regulates expression of the agrosynapine uptake and catabolism genes, it also regulates expression of this group of genes right here, of which TRAR is a member. All right, so let's consider. In the absence of the conjugative opene, that is when the bacterium is growing in a minimal medium with glucose as carbon source. All right, very natural. All right, then what you see is that ACCR is repressing this operon, and it is also repressing this operon here. So there is no TRAR, at least there's no sufficient amounts of TRAR. All right, so you can't induce the conjugative transfer system. But if you give these bacteria the agrosynapines, as though they were growing on a crown gall tumor that they had induced, then ACCR is destabilized, allows expression of the genes for the uptake and catabolism of the conjugative opene, but in addition, the expression of this operon is also now turned on. You make TRAR, and now we're back into that cycle of having TRAR, waiting for the accumulation of the acyl homoserine lactone. Once that happens, TRAR can be stabilized and dimerized, and it can activate transcription of the relevant conjugative transfer genes. And we're often conjugating. All right? I should ask, if anybody is having trouble on keeping up with me, raise your hand. My students generally throw things at me, but all right. Okay, so it seems pretty simple. All right, just the openes regulate expression of TRAR. TRAR then has to wait for the accumulation of the acyl homoserine lactone to a critical level, and then it can activate transcription. Nothing is ever simple, right? All right, so there is a complicating factor in this system, and I just want to tell you briefly about it because I think this is beginning to be seen in other systems. We have not only the activator TRAR, but we have another player in this game, and that is a small protein, 101 amino acids, coded for by the TRAM gene, which is also part of this gene system on TI plasmids. TRAM makes a protein that is an anti-activator. So the role, I won't tell you the role yet, but what TRAM can do is TRAM can physically interact with TRAR. And when it does that, it prevents TRAR from activating transcription. Okay? All right, so what's going on? Characteristics of TRAM. Well, it's a 22 kilodalton homodimer. All right, it inhibits TRAR activity. It interacts with the C-terminus of TRAR, which is the DNA binding domain of TRAR. It's induced by activated TRAR. The gene for TRAM is induced by activated TRAR. That's not particularly important for our purposes. All right. It's an 11 kD monomer, and I guess I said that already. TRAM can interact directly with TRAR. And TRAM, once it interacts with TRAR, can either cause TRAR to dissociate from DNA, or if the TRAR has not yet bound DNA and binds TRAM, it can prevent the TRAR from associating with DNA. All right, so both of those lead to an inhibition of the ability of TRAR to activate transcription. Okay, so we've done a fair amount of work on TRAM and the TRAR interaction, and we published a paper back in 2007 on modeling the TRAM-TRAR interactions. The crystal structure for TRAR bound to DNA had been solved by 2 groups, one in which Steve Winans was involved in. The crystal structure of TRAM had been determined by a group at Indiana University where Clay Fuqua was involved in this. And we had done a fair amount of genetic analysis of TRAR and TRAM looking for residues that were important for this interaction. We located them, and we were able to map them to the surfaces of these 2 molecules, and then we were able to model the interaction. We then looked at the kinetics of how this interaction takes place, and if you think about interactions between TRAR already bound to DNA and Tra-M, then what we see is the reaction goes something like this. With Tra-R bound to DNA as a homodimer, Tra-M as a homodimer, Tra-M can interact, the dimer form of Tra-M can interact with one component of Tra-R interacting at the DNA binding domain. This causes Tra-R to disassociate from the DNA, so you get this, all right, and this, and now you can get these two molecules of these two heterodimers of Tra-M, Tra-R complex interacting with each other to form the 2-Tra-R, 2-Tra-M intermediate here, and then in some ways this seems to be targeted for proteolysis, but we don't quite understand how that works. All right, we modeled this, we published it, and then a month later a paper came out in PNAS that showed the crystal structure of this, and we got it spot on. So I was very pleased with the fact that we didn't need to figure out crystal structure for this. Okay, so what is Tra-M actually doing? What's its role? Well, here's the first thing. It prevents basal levels of Tra-R from activating transcription. And so the observation is this. If you take a bacterium that has a wild-type TI plasmid and you make a mutation in Tra-M, what you find is that that TI plasmid is now constitutive for transfer. Okay? It's not constitutive for the opene that induces transfer, but it is constitutive for conjugative transfer. So we've looked at that in some detail, and the reason for this is because Tra-R is expressed at a basal level, even in the absence of the conjugative opene, and that basal level is sufficient to activate the 3 conjugative transfer operons. So Tra-M, its job is to prevent, I won't say it, its job is to prevent that basal level of Tra-R from activating transcription. Okay, that's its first job.
So I have a question.
Yeah.
Well, that happens even in a Tra-I mutant?
You will not see anything in a Tra-I mutant because you're not making any acyl homoserine lactone. If you provide acyl HL, I will show you some experiments, and actually it turns out to be a very useful tool to look at. Okay? I'll get to that, Pete. Okay, so that's the first job. Prevents premature conjugation. Okay? The second thing it does is it sets the signal sensitivity, and this is what you were getting at. It sets the signal sensitivity, and therefore it establishes the quorum. All right, and so for a number of reasons, we thought that it was necessary to make a mutation in Tra-I that was nonpolar on the rest of the conjugative transfer operon. Remember, the Tra-I gene is the first gene of the type 4 secretion operon. You knock out, normally if you had a polar mutation there, you'd never see conjugation. If you made a polar mutation in Tra-I, you'd never see conjugation anywhere because you'd be polar on making the mating bridge. Okay, so we made a nonpolar mutation in Tra-I. What we found is that if you grew this culture up and you gave it plenty of acyl HSL exogenously, the TI plasmid transfers just fine. So that's how we know it's nonpolar. Okay, but now we can ask the question, how much acyl homoserine lactone do we have to add in order to get the conjugative system to turn on? Okay, and we can do this in the wild type, and we can do it in the Tra-M mutant. All right, and so first let's look over here at this, and this has all been published. So let's look over here, and what we see in the wild type, all right, is that if we add 25 nanomolar acyl HSL, we are fully induced for transfer. If we add 250 picomolar, we are fully induced. But if we start lowering from that, 200, 100, and then 50 picomolar, then we see graded levels of decrease in conjugative transfer. So we need about 100 picomolar, about 250 picomolar acyl HSL to induce conjugative transfer in the wild type system. This guy has got Tra-M. All right. However, if we mutate Tra-M, all right, if we mutate Tra-I, all right, and also mutate Tra-M, so we have no Tra-M controlling now, what we see is that we can induce to maximum levels even with very low levels of the acyl homoserine lactone. And in addition to that, all right, we can now see this is 10 picomolar autoinducer, and now we can see some of the donors are becoming active. So TRA-M's job then, its second job, is to set the sensitivity by which the acyl homoserine lactone, by which how much acyl homoserine lactone is needed in order to induce the conjugative transfer system. Okay? All right, so I just want to make a comment about TRA-M. So this idea of anti-activators is actually quite prevalent in bacterial regulation systems. Pete's not going to talk about Pseudomonas, but there have been 2 papers that have come out in Pseudomonas aeruginosa, each one indicating that there may be a protein that acts in a similar fashion to TRA-M in the LAS-R REL-R system. Why there might be 2 independent genes that do this in aeruginosa, I have no clue, but it's a complicated bacterium, so it probably needs a complicated system. Yeah?
So I was just wondering, does TRA-M bind to also other nuclear family
no.
members apart from TRA? So
no, it only works on TRA-R, and it only works on, it is a rapidly evolving gene. All right, it only works on the TRA-R in the TI plasmid of which, or closely related TI plasmids of which it's associated. Okay? All right. Yeah?
Is it fair to say that TRA-M makes the functioning of the system switch-like? So it's like up-down?
In a way, yes. And part of that has to do with the fact that TRA-R activates expression of TRA-M about 2 to 3-fold. So you can raise TRA-M up, and then when the acyl HSL, when the opene disappears, the acyl HSL will disappear, and TRA-M can now throttle the system down. All right? I am really uncomfortable with this idea of a switch like this. And the reason is you may switch the transcription system, but you don't switch off the conjugative transfer system. The conjugative transfer system stays around for a long time. All right? In fact, there doesn't seem to be any way to degrade the conjugative transfer system. So you may turn off the transcription, but you don't turn off conjugation. Okay? All right, now, so the TI plasmid that I showed you was PTIC58. It's a plasmid in which the agrosynapine openes induce conjugative transfer, and the reason that they do is because TRA-R is a member of an operon that is controlled by ACCR, which is the repressor that responds to the conjugative opene, the agrosynapines. This is not the only opene that induces conjugative transfer. There are several others in other TI and opene catabolic plasmids that do this. And so, for example, Steve Winans has studied extensively the TRA-R of a TI plasmid called PTI-5955 or PTI-10, doesn't make any difference. The conjugative transfer system of this TI plasmid is regulated by an opene called octopene. And the reason it is because TRA-R is the distal gene of the octopene catabolism operon right here. Okay? This operon right here is very different from the operon of PTIC58 that contains TRA-R. So we have 2 independent occurrences of TRA-R being associated with an opene catabolic operon. In addition, we have looked at another opene, nopalene, that induces conjugative transfer of this plasmid right here, and it is associated with a 3rd operon. All right? So what this kind of tells us then is a couple of things. Okay, so the first thing is that this is very important that the openes control conjugation. So the way that we look at it is, in the biology of this, the regulatory hierarchy makes sense. You have the tumors produce openes, and what the openes tell the TI plasmid is that its host is in the right place for conjugative transfer, and that is on a crown gall tumor. Remember, the TI plasmid is what gives the bacterium its advantage when growing on a crown gall tumor, because it's the TI plasmid that codes for the catabolism of the openes. Okay? The quorum-sensing system, which is what the openes really regulate, does something different. It tells the TI plasmid that it's the right time to conjugate. That is, the population size is built up to a level in which, the way that I look at it, rare recipients that come into the vicinity have a higher probability of meeting a donor that has a TI plasmid turned on, and so now you have the TI plasmid conjugative transfer system activated. Okay, so the take-home message from the agrobacterium part of this, which is almost all I want to talk about, is that opines regulate conjugation by controlling expression of TRA-R, and we see this over and over again. TRA-R in these type of TI plasmids is always a member of an opine-regulated gene set. That's not quite true, but I don't want to talk about why it's a lie. Okay? The other part about this is that what this really means is that fortuitous associations of TRA-R with opine regulons have occurred many times, and that's quite clear from the DNA sequence that we have and also from the genetic studies that have been done on 3 or 4 different plasmids. TRA-R activates expression of the TRA regulon in response to the accumulation of the acyl homoserine lactone hormone. Of course, that's the way the system works. What is important here is the ecology and evolution of the TI plasmids, and that is conjugative transfer be regulated by opine availability and the population size of the donor. All right, so I want to finish this off by telling you about another plasmid of the Rhizobiales that has a quorum-sensing system very similar to that of the TI plasmid, but it regulates it regulates conjugative transfer in quite a different way. And this is work that has been done by Alan Downey's lab at the John Innes. It's the plasmid system of Rhizobium leguminosarum. So the SIM plasmid, which is PRL1JI, encodes the TI plasmid type conjugative transfer system. He would call it a rhizobium-like conjugative transfer system. It has a functional TRA-R and has a functional TRA-I. The gene arrangements are virtually the same as they are on the TI and RI plasmids. The plasmid also encodes an orphan Luxar homolog called BISAR. All right. There is a third Luxar homolog. There are more, but the third Luxar homolog that's important to the story that I'm going to tell you, which is SYNR, along with its Luxi homolog SYNI, is encoded by the bacterial chromosome. So now we have TRA-R, TRA-I on the SIM plasmid, a orphan TRA-R, BISAR on the SIM plasmid, and the SYNR, SYNI system on the bacterial chromosome. So there's a complex interplay between the SYN signal produced by recipients, and this activates the quorum-sensing system of the plasmid donor. So remember, in the agrobacterium system, the tumors induce the conjugative transfer system via the opines. In this rhizobium system, the recipients, the girls, tell the boys, turn on. Seems to be the way things work in this world. Okay, so I'm going to try to puzzle this through. This has always been a puzzle to me, but let's see if we can work this out. So here is the plasmid donor. It's got the SIM plasmid. Here's a recipient over here. These are both rhizobia. So both of them have a SYNR, SYNI complex. The recipient has no plasmid like this, so has none of these genes over here. It is making SYNI, which is this complex acyl homoserine lactone. I think, Peter, your group was the first to show that this was an acyl HSL. Used to be called fast bacteriocin. No, small bacteriocin. Small bacteriocin, right. Okay. All right. Okay. Over here in the donor, we have the SIM plasmid that has this gene system, but it also has the SYNR, SYNI system. But BISAR in the donor, in the absence of its own ligand, or in the absence of an appropriate ligand, is repressing the SYN system of the donor. So the donor is not making this hormone here. Okay? All right. In addition, TRA-R is off in this case. All right, because TRA-R needs to be activated. Well, who needs to activate TRA-R? BISAR needs to activate TRA-R. All right? So what happens then is you have the donor, and it's not doing anything, like most men. All right? Over here we have the recipient, and she's doing all the work. All right? She makes the acyl homoserine lactone that activates BISAR. Okay? BISAR now can activate TRA-R, and when TRA-R is activated now, it waits for the accumulation of the TRA-R active acyl homoserine lactone and can turn on the conjugative transfer system. So in this rhizobium system then, it's the recipient signaling to the donor, I'm here. Okay? All right, so these two plasmid types have evolved quite different strategies for mediating their horizontal gene transfer. And this, I think, is really the cool biology of this. All right? So the Agrobacterium TI plasmid quorum-sensing system has evolved to couple development of conjugative competence of the TI plasmid to a specific ecological niche. And that niche is the crown gall tumor, okay, which the bacterium has induced. All right? The rhizobium symplasmid, on the other hand, the quorum-sensing system here, has evolved to couple development of conjugative competence with the presence of a suitable recipient, all right, which is very nice. Note, the TI plasmid is recipient blind. The plasmid, once induced by chemical signals from the plant, will transfer to any recipient bacterium that happens to be in the niche. On the other hand, the rhizobium system is recipient dependent. The plasmid will only transfer when a suitable hormone-producing recipient is in the environment. However, because conjugative transfer systems are promiscuous, this does not mean that the symplasmid cannot transfer to other bacteria. It just means that the symplasmid needs a recipient around in order to turn on its conjugative transfer system. And that's the story. So I just want to tell you about the people that have been involved in this work. These people over here have done the yeoman's bit of work in my laboratory concerning the TI plasmid system. This work has been funded by the NIH, the NSF, and the USDA, and I'd be happy to take any questions.